Good evening and welcome to the Harvey lecture. My name is Sabrina Diano. I'm a president-elect of the society and also professor at Columbia University. As most of you know, this is a series of lecture today. It's part of a series of lecture that we have every year. And this is organized by the Harvey Society. The Harvey Society is a society where different university consists of different university in New York area. Each of the university has a member, a council member, and once a year we meet and we make our wish list of the people that we would like to invite, and they are the top leaders in their field. And so what we hope with this lecture series is to really bring together all the scientific community here in New York City and give it the opportunity to you, all the trainees, to get together to interact with our speakers, and of course also with each other, and hopefully come up with collaboration, with advices for your own experiments and scientific career. So the logistic for tonight is quite simple. As you may know, we're going to have a lecture from our distinguished speakers tonight. And after the lecture, there will be no Q&A, but rather an appreciation from the turnout tonight. And after that, I will invite you, and I really encourage you to follow the speaker down to the bar, where you can now approach the speaker, ask all the questions that you may have about science, about career, and also interact with one another. So tonight, it's a great pleasure to have really two pioneers in the field of lipid metabolism. Dr. Toby Walters and Bob Farese Jr. They will talk to us about the skinning of fat, mechanisms, and physiology of lipid storage. They really have been doing very incredible work in the field of lipid metabolism, and specifically how cells are able to store and mobilize lipid, and how these mechanisms play a very fundamental role in the regulation of cell function. Of course, this has a very strong implication for human physiology and also pathophysiology, specifically for metabolic disorders. And so tonight they will tell us more about this. As you can also see, normally we don't have two speakers, and this is a very special situation, very special lab, very rare in the scientific field to have a partnership, a joint laboratory. And it takes a lot of effort to have this type of partnership. That's, as I said, is not very common for a series of reasons. But most importantly, one of the reasons is because, as they pointed out, the partnerships thrives on values of respect, trust, open communication, and shared credit. It's amazing how much can be accomplished if no one cares to get the credits, and that's absolutely true in every sort of partnership, specifically in the scientific partnership. So that tells you a lot about the characters of our distinguished speakers tonight. I want to just give you a brief background of their training. I'm going to start. I was trying to put in one single slide, but of course there is so much to say about our speakers that is almost impossible. So I'm going to give you a couple of slides. So Dr. Farese Jr., he got his bachelor degrees in chemistry at the University of Florida, MD degrees at Vanderbilt School of Medicine, and then he moved for residency in internal medicine at the University of Colorado, and then eventually a clinical fellowship at UCSF, where I believe he met eventually Dr. Walther. He was first a postdoc at Gaston and then API, but then eventually in, I believe it was 10 years ago, about 2014, They moved to Harvard University, where Dr. Farese was a professor in charge in the Department of Molecular Metabolism. And only a year and a half ago, they moved to New York, where they are both at the Sloan Kettering Institute and is a professor at Weill Cornell School of Medicine. They have a lot of accolades, both of them. Just to give you a few from Dr. Farese, he got several awards from the American Society of Biochemistry and Molecular Biology. Some of these, of course, are shared with Dr. Walther. Is also was elected in the American Society for Clinical Investigation, Association of American Physicians, is a fellow of the American Society for Cell Biology. If we move to Dr. Walters, he got his PhD from his native Germany. So he got a PhD from the European Molecular Biology Laboratory and the Ludwig Maximilians University in Munich. Then he moved to UCSF for a postdoc, where eventually met Dr. Farese. And went back to Germany to be a group leader of the Max Plancks. And then a few years later, decided to come back to the States, where he was an associate professor at Yale University in the Department of Cell Biology. And eventually in 2014, together with Dr. Farese, they formed a joint laboratory, and they moved first to Harvard and then eventually here. Is the chair of cell biology program at Sloan Kettering and also professor at Weill Cornell School of Medicine. Is also an HHMI investigator. And as I already pointed before, he got several awards together, some are shared with Dr. Farese, and they are listed here. And so with that, it's really a great honor and a pleasure for me to introduce both Dr. Farese and Dr. Walters, and thank you so much for being here, and we all look forward to your lecture. Thank you.
I don't need that. Thank you very much for the kind introduction. I'd like to start with a couple remarks before we start the lecture. First of all, thank you to the Harvey Society on behalf of myself and Toby for this opportunity to present our work and for this honor. We're very appreciative and humbled by it. I also don't get a chance to give a lecture in front of family very often, and I have my daughter, one of my 5 children, my youngest daughter, Gracie Hare, and my wife, Nancy Hare. And I'd like to thank them for being here, but also thank my wife in particular, not only for support, but all of the things she's taught me and counseled me and advised me with. She's much better executive than I am. So thanks for being here. And then a few years back, a friend of mine said, you know, most people have trouble with one intensive relationship, and you have 2 intensive relationships. And so I don't get a chance to publicly thank my science partner. So I'm not going to roast him or anything. I truly just want to say thank you to Toby, because doing science together has been a great pleasure. As pointed out, we've tried to put down some of what we've learned about that. But it's just a real joy to go on an adventure of discovery with another person, and it makes it special, and certainly that has made me a better scientist, and it's made it a lot more fun. So thank you, Toby. Okay, so let's jump in. We're going to talk about lipids. We're going to talk about oils. And we're all familiar with oils as molecules of energy storage. So for example, fossil fuels power our world. So this latest figures are still we're 80% reliance on fossil fuels, and of course, fossil fuels then give rise to products that give rise to the warming of our climate. But plant oils also are very important to us as human beings, in that they provide nutrition and they provide biofuels. And animal oils or fats, depending on if they're liquid or solid, are used as energy stores by animals and in particular by human animals. And the accumulation of fat, of course, is what leads to obesity. Now, partly why oils are so good for energy storage is that they're very efficient at that. Their highly reduced carbons are energy dense. This is looking at different ways to store energy, and in light blue here are hydrocarbons, including fat metabolism as compared with sugar metabolism. And so generally, hydrocarbons provide optimal ways to store energy in a way that really doesn't require water, and that's partly why they are so energy dense. Now, why do we care about all that? Because in biology, life requires a lot of energy. So as human beings, for example, we have work to do and heat to generate in terms of energy expenditure. And kind of a remarkable statistic is that each day we have to generate our body weight in ATP. That's how much energy it takes to fight against entropic forces and create this miracle of life that each of you are. And of course, we don't generate energy ourselves. We have to take in energy. But because energy is not always available, we also have to store energy. And the primary way that we store energy is in the form of fats or oils or lipids. And this is drawn roughly to proportion for an average non-obese person in terms of how much energy is stored in fat versus carbohydrate. And a normal weight person carries around in them, for example, enough energy to run about 12 marathons. Okay, and the primary way that we store energy is in the molecule triacylglycerols or triglycerides. And so these are glycerol backbone with 3 acyl chains that are esterified to the glycerol backbone. And this, of course, creates oils, and we all know that oils and water don't mix. And this creates a fundamental problem for biology. And that is, this is an oil phase is where most of the energy is stored, and the water aqueous phase is where most of the biochemistry of life actually happens. So this has to power this. So how does nature deal with that? Well, essentially what has happened over the course of evolution is that cells have been converted into emulsions. So there are membraneless, monolayer-bound, emulsified oil droplets that provide a solution to this conundrum. And that is the cytosol of a cell can be thought of as the aqueous or continuous phase, and the lipid droplets or oil droplets can be thought of as the dispersed phase. And then, of course, to make this metastable, the oil droplets need to be coated with things that lower the surface tension, and that includes phospholipids in particular, but also proteins that act as emulsifiers or surfactants. Now, lipid droplets were actually discovered in the late 1890s by a German pathologist, Richard Altmann. And he first stumbled on this because there is phase separation in cells of oil and water, and so there's refraction and light was refracted. And this in particular is a frog embryo that was subsequently stained with osmium. And you can see all these lipid droplets or oil droplets throughout this, this collection of cells in the embryo. Dr. Altmann did quite well. He not only described lipid droplets, but also described mitochondria first and also coined the term nucleic acids, so not bad for a relatively short life of 48 years. So, lipid droplets were first described in the 1890s. Then what happened? Not much for a long time. Okay, so lipid droplets are found in virtually all eukaryotic cells as well as some prokaryotic cells. In particular, mycobacteria have a lot of lipid droplets and rhodococcus. But here, using a Badipi stain, you can see lipid droplets in yeast or fly cells or mammalian cells. And virtually any cell that we've studied, if you give them oleic acid, they will form lipid droplets. They initially had a very cool name. Okay, they were called liposomes. Isn't that cool? And unfortunately, in 1968, liposomes were invented as an artificial lipid droplet made in a test tube, and that kind of usurped the name. And so lipid droplets were a long time in search of a name, and they've been called oil bodies or lipid bodies and various other names, but the field, at least in the mammalian field, has kind of arrived at lipid droplets as the terminology. And as I said, not much then happened for more than about 100 years. But then if you look at the citations for lipid droplets, you can see that, again, not much happening, but then right around 2006 or 2007, there was a big uptick in the number of papers on lipid droplets. And this largely coincides with systems-type approaches that were brought to the field. Genetic studies that identified genes that govern lipid droplet biology, proteomics that identified proteins on lipid droplets. And what that unmasked was a rich biology that subsequently has been studied as sort of a new frontier of cell biology. And this is about when Toby and I also started working on things. And we were fortunate enough to be involved in the opening up of this new frontier. Okay, so this is a schematic that shows lipid droplets. So they are monolayer bound, but essentially they don't have a bilayer membrane, so they're membraneless organelles. And this is an electron micrograph of a lipid droplet from a hepatoma cell, where you can see a monolayer of phospholipids coating the neutral lipids inside. And they're decorated by various proteins, as I mentioned. Typically, lipid droplets are, in most cultured cells, are about 300 to 500 nanometers in diameter. However, in human hepatocytes and a fatty liver or adipocytes, they can be microns or tens of microns in diameter. And they're often found in close association with other organelles, the endoplasmic reticulum, which is, you'll see, is where they arise from, as well as mitochondria and in particular. And they're very efficient ways to store energy. So if you do a back-of-the-envelope calculation for a typical lipid droplet, it can hold 21 million triacylglycerol molecules, roughly, or the equivalent of 23 billion ATPs. So these are very potent energy stores in all of our cells. Okay, so with that background, what Toby and I are going to do here tonight is we're going to try to answer 3 questions best we know for the field at this point. How do cells generate this oil phase? How do they get substances in and out of the oil phase? And how have organisms evolved to utilize this oil phase in physiology? And then what can go wrong in that? So we'll give you a sampling of some of the biology that have been uncovered mostly in the last 15 to 20 years. And I'm going to start by talking about this question in the first half of this, and then Toby will take over and talk about some of how proteins get in and out of oil phases in the physiology. Okay, so the first question is then, how do cells make oils? And this history also goes back to the 1800s when triacylglycerols were discovered, but the biochemical pathway was really unraveled in the 1950s, and in large part by Eugene Kennedy and co-workers, who unraveled a lot of glycerolipid biosynthesis, all the phospholipid biosynthesis pathways, but also how they branch off into neutral lipids like triacylglycerol. And so what happens, or what they described here in this paper in 1960, is that in microsomes, or fractions from the endoplasmic reticulum, fatty acids activated by covalent bonds to a CoA molecule and diacylglycerol can come together through the actions of enzymes to form triacylglycerols. And so that activity was described then. Then not much happened again for quite a long time. People tried to purify these enzymes that make triacylglycerols, but that initially was not very successful. It turns out at least one of the enzymes is extremely hydrophobic and difficult to purify. But about 40 years later, my laboratory, actually before Toby and I met, serendipitously uncovered the gene encoding DGAT1. And we weren't looking for it, so it's a great lesson in science. We were looking for other things, but we found this and we recognized it and quickly turned the ship in another direction, basically. And so we cloned the gene that for a diacylglycerol acyltransferase that makes triacylglycerols in the ER. It's now known as DGAT1. And shortly thereafter, I was called up by folks at Calgene who were also looking for enzymes that made oils. And they said, well, we got a DGAT in a fungus, and it doesn't look like DGAT1. And it turned out that this had 7 orthologs in humans, and the primary one that they had cloned is now known as DGAT2. Other members of the family make other neutrolipids, such as diacylglycerols or wax esters. Okay, so now you have 2 enzymes that essentially co-evolved to make triacylglycerols in oil. They don't share any sequence homology, and they basically arose separately with the same biochemical reaction. Fast forward another 20 years or so, we did a lot of studies on DGAT1, but with respect to its biochemistry and physiology, But then sort of a dream come true, if you've cloned one of these genes, is that you'd love to really understand how it works. And thanks to the miracles of cryo-electron microscopy, single particle, we were able to get a structure, a model structure for the DGAT1 enzyme. Our lab did this coincident with a lab at Baylor at the same time, and the structures are virtually identical. Now, the DGAT1 enzyme is a dimer, and it has this beautiful butterfly shape with the N-termini that reach across to the other side. And what we really learned from this is that this is how we envision that it sits in the endoplasmic reticulum membrane. And what we learned is that is basically what we can speculate or guess about how it catalyzes oil or triglyceride formation. And that is all the enzymes of this family that DGAT1 is from, which is the M-boat family, membrane-bound O-acetyltransferases, have conserved histidines. And for DGAT1, it's histidine 415. And we could see that that sits deep buried in a hydrophobic pocket within what we think is the plane of the membrane. And so what we learned from this is that the reaction probably proceeds somewhat like I'll show you, with access to this catalytic channel from the cytosolic side, as well as from the plane of the membrane through what we call a lateral gate. And so what we envision is the substrates from this reaction enter through different mechanisms. Fatty acyl-CoAs are likely in the membrane if they're long. Here's the CoA handle and the fatty acid. And they get inserted into the catalytic chamber, and here's the catalytically required histidine molecule. Whereas diacylglycerol, the acyl-CoA acceptor, comes from the plane of the membrane and enters the enzymatic center that way. Here, catalysis happens, which this histidine is involved in. Triacylglycerols are formed along with CoASH. The CoASH is released into the aqueous environment, and we speculate that, and there's perhaps some molecular dynamic simulation data that suggests that the oil then is released into the plane of the membrane, and really it has to be released into the plane of the membrane. So we have a cryo-EM derived structure for DGAT1. That's a dimer with two catalytic centers to this dimer, as denoted by these stars. We do not yet have a structure of experimentally validated structure for DGAT2. But DGAT2 has an alpha-fold structure. It was actually cited in the original alpha-fold publication. And it looks quite different, as you might expect. And so DGAT2 seems to have a hairpin that embeds it into the membrane, but its active site sits up here at the surface of the membrane. So presumably, it catalyzes this reaction to the surface of the membrane and releases the product into the membrane. Okay, so that's a bit about how do cells make oils such as triacylglycerols. Of course, they make other oils like sterol esters and wax esters and so forth, but much of the paradigm is similar for those other nutrilipids. The next question is, how do they get the substances in and out of the oil phase? And so this is the question that really started our work together when I sat down on sabbatical next to Toby, and he said, "What are you doing?" And I said, "Well, somehow cells make these really cool organelles called lipid droplets." You can see a video of a cell making lipid droplets here. This is over many minutes adding oleic acid. And you see this is the cell, and you see throughout the endoplasmic reticulum, these small droplets are made in a regular manner, and they're rather uniform in size. And this was like, wow, how does that happen? And you go to the textbooks, and there's nothing. Okay, so that's an opportunity, right? So how do we figure out what's going on here? And that really was the catalysis of the beginning of a lot of our work together. So we know that triglycerides are made. They're probably released into the plane of the membrane, and then somehow they make a droplet from the endoplasmic reticulum. What happens there? Now, one of the critical things that was known at this time was from the work of Jim Hamilton and Don Small in 1983. They had shown in vitro in test tube experiments that if you take bilayer membranes, the bilayer can solubilize up to about 3 mole percent triglyceride within the bilayer. But above that, it will nucleate, phase separate, and form a lipid droplet spontaneously. So it was our hypothesis that there must be genes or proteins involved to organize this process so it just doesn't happen randomly all over the endoplasmic reticulum. And it must happen, we envision, at a concentration lower than 3 mol% to prevent the spontaneous emulsion formation. Okay, so what might those proteins be? Well, it turns out that the protein called sapin, named after Dr. Sape, who described this congenital lipodystrophy in the 1950s. So Berardinelli and Sape described congenital lipodystrophy, and this sapin took the name from Sape. And basically, this protein was involved, was identified as a cause of congenital lipodystrophy around 2001. And what sapin is a protein that's in the endoplasmic reticulum and highly conserved in these 2 transmembrane domains and this luminal domain. And it had mutations that gave rise to lipodystrophy. So that was a clue, but nothing really happened until, as I mentioned, systems-type approaches and genetic screens allowed people to start identifying the genes and proteins that were really involved in lipid droplet biology. And so a series of screens were done, initially in yeast, but also in worms, flies, humans, et cetera, human cells. And this is an example of that. So a number of years ago, we did a screen where we're looking at things that affect lipid droplet morphology in cells. We have over 300 genes that cause all kinds of different types of lipid droplets, whether they're small, large, dispersed, et cetera. And the #1 hit in our screen is this gene, Bernard Sape, congenital lipodystrophy 2, BCSL2, which encodes the sapin protein. And you can see here that, it's a pretty obvious phenotype, whereas instead of all these small droplets, you have these larger droplets. So we and others have been studying sapin and what does it do to help make lipid droplets. And so what you can see here, these are fly cells where we've added oleate, and this cell is forming lipid droplets. It has some preformed lipid droplets, and then you can see, like I showed you before, there's a regular formation of droplets in the cell over time. If you knock down or knock out sapin, you see a phenotype that's very different. You see a disorganization of lipid droplet formation, and you see tens of little tiny small droplets, and then some really big droplets. And I'll just tell you that the big droplets result because the little droplets are not coated with enough surfactant, and so they end up coalescing to form large droplets. And electron micrographs revealed in these cells that these droplets were about half size than normal. They were always localized close to the endoplasmic reticulum, and they had completely budded off from the ER. So they had detached from the endoplasmic reticulum, as you can see here, in all the instances that we could see. So using, again, technologies that have emerged in recent years, CRISPR, we were able to tag sapin and look at sapin in Drosophila cells. You can see that it forms punctae. And these punctae move around the tubular endoplasmic reticulum. And here you can see in a human cell, tagged sapin, endogenously tagged sapin does something similar. So you see these sapin punctae that sit in the endoplasmic reticulum. And as you form lipid droplets, essentially what you see is that every lipid droplet has one sapin punctae near it. And we and others have consistently found this one sapin punctae per lipid droplet is what you find in normal cells. Now, again, thanks to cryo-electron microscopy, we now have a picture of what sapin looks like. And what you can see is that it is a cage-like structure of individual monomers. And the monomers can be 10 or 11 or 12. And the transmembrane domains sit and cross each other, as you can see there. And if you peel back, you see that there's a cage in the middle. And we don't know exactly how this works, but of course we can hypothesize how it might work. And what we think, or our current model, is that triglycerides in the membrane can get access into the middle of this cage-like structure. And that cage will exclude phospholipids and exclude other proteins and sort of form a chamber, and that chamber allows the hydrophobic triacylglycerides to initially come together, nucleate, and then begin phase separation that matures after that. Now, this can be tested experimentally in molecular dynamics simulations. This was done by Si-Young Kim, a collaborator of ours, and Greg Voslab from the University of Chicago. And here is a vesicle with 2% triglyceride in it. And you see under the conditions of this molecular dynamics simulation, there is no phase separation or lipid droplet formation. However, with the same conditions now with sapin structure placed into this vesicle, you can see that this does, in about half the instances over the course of this time course of the molecular dynamics simulation, result in phase separation and the beginning of a droplet form. And when this happens, the other thing that Seung could see is that the sapin oligomers actually open up, so that cage structure tended to open during the molecular dynamics simulation as the droplet grew. And essentially what we think is that it forms then a neck, and that neck keeps the droplet attached to the endoplasmic reticulum. The other thing that we found from the structure is that if you look at it in a ribbon-type model, what you see is an alpha-beta sandwich folds in the luminal domains, but this ring of hydrophobic helices that sits at the center. And this ring, we think, is embedded into the membrane of the ER. And Ji-Yun Chung, when she was in our lab, did a pull-down experiment with or without that hydrophobic helical ring. And one of those really rewarding experiments where she found only one protein was different between these two. And it was a protein called TMEM159 that we call now lipid droplet assembly factor number 1, or LDAF1. And what was able to do was to look in cultured cells, knock in endogenously tagged sapin and LDAF1, and also a marker for lipid droplets, PLIN3, that's a very, very sensitive marker. And what she could see is that everywhere where lipid droplets formed, there coincided sapin and LDAF1. And this was sort of the first demonstration that actually lipid droplets form at this complex, and what we now call the lipid droplet assembly complex. We're in the process of trying to get a structure of the whole complex with both sapin and LDAF1. Pedro Carpio-Malia in our lab is working on that, and this is the latest progress that he's had, where we see the sapin monomers here with transmembrane domains and the luminal domain and the hydrophobic helix. And then we see all these densities here that are an inner ring of this LDAF1, so it's somewhat as we envision. And if you look at what happens with sapin and LDAF1 in purple, sapin in green, they co-localize, but then as lipid droplets form, LDAF1 coats the growing lipid droplet and sapin stays at the endoplasmic reticulum. And for those of you that are old enough to remember Jiffy Pop popcorn, it kind of looks like that because you have this sapin ring with LDAF1 in the middle here, many monomers of that, and then as the droplet grows, it's coated by LDAF1, we think, that comes from the endoplasmic reticulum. It turns out that LDAF1 is not essential. You can still form lipid droplets without it, but you see you form less lipid droplets and larger lipid droplets, and for a given amount of triglyceride, you form fewer lipid droplets. So we think it's a factor that enhances the ability of droplet formation, but is not absolutely required. And so that leaves us with this model. This is our current model of how lipid droplets are formed. And this modeling was done by Janet Iwasa, who's sort of an award-winning scientist and graphic illustrator. And so this is how we envision the process. If we zero in on the endoplasmic reticulum, we have fatty acids and diacylglycerol. These are the DGAT enzymes, DGAT1 and DGAT2, that are catalyzing triglyceride synthesis. Triglycerides diffuse within the membrane. They nucleate within this chamber. This is LDAF1. That sapin, we think, opens up. And then as this grows through a ripening type process, you get a mature lipid droplet that's anchored to the membrane through the sapin oligomer complex. Okay, so as we do in these lectures now, I depart, and Toby will take over with the second part of the lecture.
Thank you, Bob. I want to go back for a second to this very simple model, biochemical reconstitution here of what's happening. And we could liken this to some degree to a piggy bank of energy, metabolic energy in the cell, where we have energy deposits, and this is only useful, of course, if you can deposit and withdraw energy at any given time. And in the course of physiology, for instance, in the liver, there's dramatic changes, and cells not necessarily make always new lipid droplets, but they can mobilize energy resources from existing lipid droplets. For instance, here, this is a histology slide of a liver on either a regular diet or a high-fat diet, and you can see dynamically how there's a lot of lipid droplets formed that through the course of this varying availability of metabolic energy grow and shrink. But this generates a massive problem. Like, how do you actually do this? And this is solved by a number of proteins that localize to the lipid droplet surface that Bob already mentioned to you. Now, these proteins have to localize at the interface between oil and water and mediate essentially all the communication and exchange between proteins. This is a non-trivial feat for a simple reason, and that is that this interface here is highly denaturing to proteins. Some of you, as biochemists, may remember that, of course, proteins are amphiphilic. They have hydrophobic and hydrophilic parts, which drives their folding normally. But also, this provides a principle that we use sometimes to isolate things, because when we extract a tissue, for instance, with an organic phase, we can isolate RNAs and things of that sort, polar things, in one phase, and all the proteins become denatured at that interface because it is trapping amphiphilic molecules. And so really, this generated a problem for evolution, and many proteins actually can sort of do pretty good until they see the oil phase where they get denatured on the surface. But it has turned out that there are a large number of proteins that are specific to lipid droplets that have mastered this feat to live at that phase boundary between the oil and the water. And it was a breakthrough discovery by D. Landas and his group that there are specific proteins that are really only found on the surface of lipid droplets and not elsewhere. And proteomic screens in a number of laboratories, including ours, have identified several hundred proteins that are specific to that surface. And they include proteins that you might expect that are involved in the synthesis of oils, triglycerides, and it includes proteins that are involved in the withdrawal of energy through lipolysis, for instance. I don't have time today to talk about this, but we also found a whole bunch of surprises. Be happy to talk about that. There are transcription factors that are regulatory. There are enzymes for which we have no idea what they do and why they're there. Now, how do these proteins get there? And this is a non-trivial feat because the cell is a very crowded place. And particularly, lipid droplets are, of course, membrane-less monolayer organelles embedded in the network of membranes known as the endoplasmic reticulum. As you know well here at Rockefeller, of course, the signal hypothesis has predicted how things go to places in the cell. And the problem is, for lipid droplets, there is no import machinery that reads those signals. There are no specific lipids, for instance, such as phosphoinositides, that attract specific types of proteins. And for all intents and purposes, if you're swimming out here somewhere, this surface should look identical than the surface of lipid droplets. And so it is really a question of how proteins have mastered to specifically detect the lipid droplet and accumulate there. Now, it turns out there are two solutions to this problem, it appears, at least. And one of them we call cytold cargoes for cytosol to lipid droplet. In this pathway of targeting proteins to lipid droplets, proteins are initially synthesized in the cytosol, and they contain amphipathic sequences that directly bind the surface of a lipid droplet. This includes a large number of proteins. Some of them are important for lipid metabolism, others are regulatory. And what it turns out, how they read out the surface is that despite the fact that the lipid composition from the cytosolic site here appears similar, there is very important differences between those surfaces that are a consequence of the underlying oil phase. In particular, this is a depiction from a simulation. The presence of triglycerides, here depicted in green, pushes apart to some degree the phospholipids. This is known as packing defects. And these packing defects are similar to what would happen if you really stretch a membrane and allow insertion of proteins that can detect these hydrophobic surfaces on the lipid droplet. An experiment that illustrates this was done by Colleen Prévost in the lab, where she used a giant unilamellar vesicle and into which she incorporated triglyceride to come up with an in vitro system mimicking lipid droplets. And then she can vary the tension and hence the amount of coverage of that lipid droplet up here. And we can at the same time monitor binding of proteins. And here's the experiment, and you can see that as we increase the tension and therefore increase the number of packing defects, proteins bind to the surface of lipid droplets. And this is one principle to drive specificity, because now proteins have sequences that are specifically evolved to read out the packing defects, and then they fold into helices that have one hydrophobic and one hydrophilic layer and are specifically adapted to that surface. Incidentally, such a process that is folding dependent essentially renders this process irreversible. And it turns out that in order to come off a lipid droplet, a different process is required. A second pathway initiates with synthesis of proteins in the endoplasmic reticulum and then leads to accumulation of proteins on lipid droplets. This includes a whole number of cargoes that are important for lipid metabolism and physiology. And we know that they go to lipid droplets because we can localize them with antibodies here with a gold label. This is a time's up signal when your pointer runs out of battery. When you can see that this decorates specifically the surface of lipid droplets, but not the endoplasmic reticulum close by. So how does this work? Of course, we know that proteins can't just hop in and out of membranes, and it turns out that for one of the model cargoes that is known as GPAT4, which catalyzes the first step in glycerolipid synthesis, a short sequence is sufficient to drive the accumulation of lipid droplets. Now, the problem in analyzing this process is that lipid droplet formation takes hours, and the targeting takes seconds. So Arda in the lab recently set up a system that allows us to synchronize this, and suffice it to say, we can anchor proteins in the endoplasmic reticulum, and then using a streptavidin binding peptide interaction, and then release them at a specific point and analyze this. And what you can see is that in this system, synchronously, you can release the cargo, and everybody goes from the ER very quickly within minutes to lipid droplets. Because of that synchronized way, we are now able to analyze this process using single molecule fluorescence. So every dot you see on this here is a single molecule of a protein that goes to lipid droplets. And we can follow this with high time resolution. And you can see, that the proteins explore the endoplasmic reticulum network and then accumulate on lipid droplets that appear as these hairballs as we track this with very high spatial density. I won't have time to tell you all the things we've learned from these experiments, but suffice it to say that one thing is that we learned is that apparently the proteins that go onto lipid droplets do so directly at the sites of their formation, the specific SAPE and complexes, and that the diffusion breakpoints where they enter the lipid droplet is always right there where SAPIN is localized. So this leads us to this model in which at least this cargo here, this little hairpin, actually can go through this complex and access the lipid droplet directly. Now, this is nice and easy, and this is what you would expect, unless someone does an experiment. In this case, this was Florian Wilfing, a graduate student in the lab a few years ago, who sat for hours in front of the microscope while nothing was happening, in this case, to the full-length protein. I still don't know why he waited 5 hours, but he did. And then within 5 hours, nothing happens, and then within a few minutes, one lipid droplet acquired a whole bunch of protein, in this case, the full length. And that's a mystery. It's like, why does nothing happen here? What's the trigger here? And it turns out that because cells don't always make lipid droplets, it appears that they synthesize multiple connections with the ER after their formation. This is visualized here in an electron tomogram, and here by a fluorescence image. This is an interpretation of that image in which we see multiple connections that are formed between the ER and the lipid droplet. And we believe that this is the route for these proteins, because maybe when you're large, in a large cargo, you can't go through this complex, which is indeed the case. We have an experiment that I'm not going to show you, but SAPIN blocks many, many cargoes to go through here, and it allows some. We do not yet understand either how this is achieved mechanistically or what's its significance, but instead cells have machinery that will establish secondary structures and bridges between the ER and lipid droplets that allows many cargoes to access lipid droplets even after their formation and independent of the sapien complex. Now, those two pathways, in our mind, explain that there is a road between the ER and lipid droplets. But just because there is a bridge over to Brooklyn doesn't mean everybody has to run over there and we all meet there, right? So what's actually the driving force that leads to accumulation? Now, we don't know the full answer to this, but why would those proteins not just equilibrate between those two, but really accumulate on lipid droplets? The answer to this lies in the specific hydrophobic sequences that these proteins have evolved, which are peculiar because they have a couple of unique features. So one thing is, like, we can scramble that sequence and all of a sudden you can still be in the ER, but the proteins won't accumulate anymore. And particularly, there are large hydrophobic residues in the middle of a hydrophobic segment that are required to lead to accumulation. This is highly unusual. For those of you who study membrane proteins, you will find that usually tryptophans are on the interfaces between the cytosol and the membrane and not within the membrane bilayer. And what the current model is based on experiments and simulation is that within the bilayer, these proteins are inserted in a configuration that is thermodynamically unfavorable because it places large tryptophans in the center of the bilayer, and that when these proteins move to the surface of lipid droplets, they swim up, the tryptophans become interfacial, and that there's a lower energy, and therefore those proteins accumulate. We've shown this for the protein GPAT4, but I also have made similar observations with a number of other proteins. Now, in the last piece of this, I want to convince you that studying basic cell biology can have impact on human physiology and understanding of disease. Now, Bob already mentioned that, of course, the accumulation of lipids is the hallmark in the development of obesity. When humans accumulate massive amounts of fat in adipose tissue, that's what we know in obesity. But of course, I want you to take home also that lipid droplets can form in pretty much every other tissue. For instance, in the intestine, after a meal, temporarily lipid droplets are formed to store triglyceride before they're being shipped out or metabolized. And consistent with that, when tissues are faced with an excess of fatty acids, depending on where the fats accumulate, different types of pathology emerge, particularly, for instance, fatty liver disease or cardiomyopathy and other diseases driven by lipid accumulation. Now, if it is a problem to accumulate fat in lots of different tissues, and we know the enzymes that do that, we should just make inhibitors, right, that block the synthesis of the triglycerides, and the problem's gone. And that, in fact, looked like a really good strategy, because it turns out that when Bob and colleagues knocked out DGAT1 in mice, it has a fantastic phenotype. These mice are lean, they're resistant to diet-induced obesity, they live 30% longer, they're just fantastically healthy. So that's great, right? So this was really inspiring and has led to the development of very good inhibitors by pretty much every major pharma company. And of course, the problem was that for a while this wasn't tested in humans. And it turns out in humans, when these inhibitors became available, it overlapped with observations we had made with identifying patients that have mutations in DGAT1. And it turns out that this leads to a very severe congenital diarrheal syndrome that is a severe fat-dependent diarrhea in kids, pediatric syndrome. And the first patients that were identified actually succumbed to the complications from these conditions. And consistently, this was observed in the trials observed with this. So it turns out the difference between humans and mice, we believe, is that humans only express DGAT1 in the intestine, and mice express DGAT1 and 2. And what is common to a lot of the diseases that are associated with the overaccumulation of fat is that the capacity becomes overwhelmed to store fatty acids in lipid droplets. As a consequence, excess toxic lipids accumulate, and this leads to adaptation or cell death. In the case of the gut, when a lot of fatty acids come in and we only have DGAT1 and the patients now do not have functioning DGAT1, this pathway doesn't happen, enterocytes die, and this severe disease occurs. And consistent with this, there now is a large list of genes that are associated with particular pathologies, depending predominantly where the proteins involved in lipid droplet formation and metabolism are expressed in the body. Many of them are metabolic diseases, and some of them appear as neurological diseases. I'll be happy to talk about that afterwards. How about DGAT2 then? Like, can we do something about the other drug? Well, this is the opposite example. If you knock out DGAT2 in a mouse, the mouse is dead. That does not generally engender a lot of confidence in this protein as a drug target. Nonetheless, we started to do experiments, in particular Nina Klochowski in the lab, to specifically inactivate this protein in the liver. And what we found is that when we take a mouse, particularly males, and feed them a very large amount of fatty acids, the normal mice, control mice, accumulated a lot of fat in their liver, known as hepatic steatosis, whereas the knockouts did not. And moreover, we did not observe any adverse consequences really in these. And this is really, we believe, could be an important result because, as you may know, currently 1 in 4 people in the globe, not just in the US, but in the whole world, suffers from fatty liver disease, which is considered to take over as one of the main pathologies for the liver. And so the question is, maybe DGAT2 inhibitors could be a good drug to inhibit triglyceride synthesis. And so particularly for the indication of fatty liver disease. And so it turns out what I just showed you, hepatic DGAT2 deletion protects actually from fatty liver, and this has been replicated in other studies. And this has motivated particularly Pfizer, but also other pharma companies to start human trials on inhibitors for this, which has markedly reduced liver fat. Here's an example for a study where multiple doses were given over a relatively short period of time. I believe it's 2 weeks. And you can see a marked reduction in triglyceride. And the intriguing part of this is, like, if what I told you before about lipotoxicity is true, this should also not work. But it turns out in this case, the inhibitors do 2 things. They block the triglyceride synthesis, and they also turn down lipogenesis. They turn down the synthesis of more lipids. And that turns off the spigot, if you will, and prevents the accumulation of more lipids. And this has allowed these drugs to move forward to now phase 2 and 3 clinical trials. So we're hopeful that these insights might lead to better therapeutic approaches. Now with this, I want to just wrap up a little bit. Bob and I tried to show you today our insights about fat, how fat is made, how lipid droplets are formed, and how proteins get to lipid droplets. We also wanted to share with you that we believe those basic insights into foundational biology can be useful. And all of this is only possible because we've had amazing people in the laboratory. Some of them are here, actually, surprisingly. Thank you. And I don't want to go through all the names of our amazing trainees and former colleagues. I want to take the opportunity also to just speak for a moment. I also have some family here. My wife, Mara, who's been an amazing companion since we met in Bob's lab. We'll talk about that at the bar. And also, as a happy coincidence, this I didn't think was going to happen. My mother is here, who is visiting from Germany, and I don't think has heard me or Bob give a lot of lectures. I want to thank you both for being just amazing and supportive all the way. It's also one of the reasons why we're here is, I think, because Joan has brought us to New York, and I want to thank you for all your support in the last 1.5 years or 2 years on this journey in the next chapter. I feel like I don't need to thank Bob because he knows. But I love, when we started working together, our major inspiration and aspiration are Mike Brown and Joe Goldstein. And it's not, I don't want to put us in the same universe, right? But it is something to aspire to. And there is a wonderful interview where they ask Joe Goldstein what it takes to have a scientific partnership. And there's a near endless list of things that you need to do. And in our case, that's Bob. He thinks about these things deeply. And then they ask Mike Brown, and I don't want to compare myself with him, but the answer is something I would have given. They ask Mike Brown, what does it take? And when they ask me, what does it take? He just says Joe Goldstein, and in our case, it's just, you just need a Bob. It's amazing. Thank you so much. It's been an amazing journey. Thank you very much.
Thank you very much for this incredible seminar. And now it's my pleasure to introduce Dr. Ushma Neal. She's the Vice President for Scientific Education and Training, Memorial Sloan Kettering Cancer Center, for her precision remarks. Thank you.
Hello, everyone. That was a spectacular lecture, Bob and Toby, and I just hope you will still be talking to me tomorrow. Okay. Hello, everyone. I'm Ushma Neal. You might be wondering who I am and maybe why I was chosen to give this appreciation. Well, I wear two hats, one where I am the Vice President of Education across the street at Memorial Sloan Kettering, and I am a colleague to both Bob and Toby, but I'm also editor-at-large at the Journal of Clinical Investigation, and before that, for 9 years, I was the executive editor of that journal, and through that job, I got to meet a cross-section of different scientists. And so though I spend a lot more time with Toby these days on administration, our story tonight begins with Bob. So I met Bob at a dinner party at a Keystone meeting in February 2008 when I was pregnant with my second child, who will feature in the slideshow later on. And I've been able to measure our friendship alongside my ever-growing now 16-year-old son, who is now taller than me. So I went back through, in preparing for this appreciation, through 16 years of emails with Bob. And I actually found one from the Harvard-based cardiovascular researcher Jorge Plutsky that mentioned Bob and that very dinner party. And he joked that I was the world's worst dinner guest as I sat at one end of the table and talked to Bob and Bob alone for 2 hours. So what can I say? Bob was an intriguing conversationalist back then and has remained so for the last 16 years. So if I could please have the slides. So anyways, for tonight's appreciation, I thought I would do a play on how it started and how it's going, the Bob and Toby edition. So I hope you will enjoy this half as much as I enjoyed putting this together. Okay, so I begin with how it started. This is a poster from 2005 of when they first met and they were giving lab meeting together. And how is it going? Well, I think you can see that they are still having a very good time together. I believe in this picture, Bob is impersonating Howard Hughes in celebrating when Toby was named an HHMI investigator. So let's begin with Bob and how it started. So here's a teenage Bob growing up in Florida. His father, Robert Farese Sr., was a physician scientist who practiced as an endocrinologist and studied how insulin acts in obesity in his lab at the University of South Florida. I do admit that at one point as an editor, I sent a review request to Dr. Farese Sr., to which he replied, "I think you meant to ask my son." Anyways, Bob had a great role model to follow en route to a bachelor's degree in chemistry at the University of Florida, followed by an MD at Vanderbilt, before moving to Denver for his internal medicine residency, wherein he also had a year as chief resident. He decided to follow the inspiration of his father and moved to San Francisco in 1989 to take up his endocrinology clinical fellowship, alongside a few years as a postdoc in the lab of Steve Young, studying apolipoprotein B. During this time, Bob created the first ever knockout of an apolipoprotein gene. Okay, so we are on to the how it's going phase, with Bob as a young assistant professor here in a charming sweater. Okay, for you younger scientists in the audience, I must try and convince you this was the height of fashion back then, so no shade allowed. All right, so during this phase, Bob made discovery after discovery in the realm of cholesterol ester biosynthesis and physiology, including discovery of the genes encoding and description and function of monoacylglycerol acyltransferase and the diacylglycerol transferases, DGAT1 and 2, that he just described for you, all of which are crucial enzymes in the synthesis of triglyceride. So during the course of study in this field, and we've already heard about Brown and Goldstein once, Bob got to know the OGs of scientific partnership, Joe Goldstein and Mike Brown. So Bob got some advice from Mike to help reinvigorate his science. Quote, "One idea is to take a sabbatical in a lab that is using technologies that are totally different from the ones you use. For example, you might study a problem in cell biology, membrane biology, or gene transcription. Then you could return to lipid metabolism with some fresh ideas," which led him to the lab of the scientific unicorn, Peter Walther, who has taught us much of what we know about the unfolded protein response and cellular quality control. It was during Bob's sabbatical in Peter's lab that he met a postdoc named Tobias Walther. From a published interview, quote, "Toby was a very rare kind of a person. When you do a sabbatical, many people in the lab ignore you, but Toby, without hesitation, said, 'I'll help you, let's go.'" And in another interview, quote, "One day, Bob sat down next to Toby, then a postdoctoral researcher in the lab, and intellectual sparks flew." I didn't write that, right? Okay. Okay, so let's go back to how it started, this time the Toby Walther edition. I was expecting more laughs than ahs, okay. And yes, I have conspirators in acquiring images, and they may or may not be in the audience today. But please, Toby, direct your ire at me if you are displeased with this. I just could not choose between all these fabulous images of a young Toby growing up in Germany, a fan of phones, skis, Oompa bands, and pacifiers. And in a mini How It's Going, I can't tell you how many pictures there are out there of Toby skiing. It's impressive even if he left the pacifier behind. All right, so Toby managed to swing an exchange as an undergraduate student at the Southern Methodist University in Dallas, but finished his Diplom Biologe at the Ludwig Maximilian University in Munich. He then stayed there for his PhD, working at EMBL, where he identified factors of nuclear pore assembly. He then, as we have now noted, joined the aforementioned Peter Walther's lab for his postdoc, work that resulted in two spectacular papers, one on the identification of novel cortical structures in yeast called isosomes, and the other, with Bob, showcasing the first systematic RNAi-based screen in mammalian cells for proteins implicated in lipid droplet dynamics. So given the strength of his abilities to bring new, dynamic, and complicated screening approaches to the study of cellular structures, and specifically lipid droplets, he was offered a group leader position at the Max Planck outside of Munich. During this time, he took a great liking to proteomics and published a few citation classics, including the first complete and quantitative proteome of any organism, budding yeast. And there are not a lot of pictures of this time in Toby's life, by the way. I tried really hard. All right, so his ongoing focus on lipid droplets, as well as his cutting-edge approaches to understanding them, landed him as an associate professor at Yale for 4 years, using sophisticated molecular genetics and dynamic imaging approaches to ask questions about molecular traffic and communication between lipid droplets and the ER. So in their CVs, it's beyond obvious that when these two met, they fell in scientific love, and that even though they were in different labs, they continued to work largely with each other. So they decided to go all in and start a joint lab in 2014. And this is the time when their work really exploded, at least by my estimation in looking at their CVs, with the discovery of protein machinery, mediating orderly assembly of lipid droplets from ER exit sites, to the use of cryo-EM to fully elucidate a molecularly precise model for lipid droplet assembly, and allowing them to put together what we so brilliantly heard them talk about for the last hour. So you've now seen this for the third time, but in an article in my beloved JCI, they wrote about how their work isn't just additive but multiplicative, but I'll use a different quote than Sabrina did. "We have enjoyed our scientific journey together immensely and can barely remember what science was like in the years before. Without a doubt, we have become better scientists because of our partnership." Now, I told you there would be several acts to this play, right? And I also started by telling you I'd show you a picture of my then infant son, who is now taller than me. And for any of you who have had children, you know that the minute anybody asks or even doesn't ask, you send pictures of your children, and especially when they are dressed up for Halloween. And this was the child I was pregnant with when I first met Bob. So I had sent this picture to Bob of my two kids dressed up as bats, to which I received this picture in reply. I gotta say, that tattoo looks a little bit like a membrane with sapin in it, doesn't it? Okay. So recall, this was the time during which I was the editor of a very august journal, and I had at the time a penchant for making images into fake covers. So I could not help myself, and Bob was the cover model for the January 2009 issue of the JCI. But I am happy to report in this version of How It's Going, Bob and Toby would get the cover again. All right. So when we began, I told you I was having a lot of fun going back through 16 years of emails with Bob. And in our last vignette of How It Started and How It's Going, I wanted to show you one email chain, this time focused on MSK. So in the first of the How It Started, from May 2019, when Toby gave a talk at MSK, I noted I could barely get in the door and remarked, "Who knew a bunch of cancer biologists would have such an appetite for lipid droplets?" To which Bob replied, "Glad to hear our work is of interest. Lipid droplets are showing up all over, including in cancer." And so I replied, "I hope Toby enjoyed his day here. The department that invited him is full of terrific colleagues." Happily, he agreed, and it might have taken us a few years, but in September 2022, we at MSK were able to recruit and welcome Toby and Bob to that very department at MSK, with Bonus Mara Minetti leading the proteomics efforts at MSK. So through those 100 emails I exchanged with Bob, there was a very common theme: his family, some of whom are here tonight and I know played a huge role in getting Bob to where he is today. And the same goes for Toby. In fact, the first meeting I took with Toby when he got to MSK was to discuss New York City schools for his girls, and always with the brilliant Mara, who I may or may not confirm was my main co-conspirator tonight. All right. Lastly, I wanted to show this photo of the old guard and the new guard of scientific partnerships together from an event in 2018. I wrote to Mike and Joe this weekend to ask them if they happened to have a version of this picture where any of them were looking at the camera. Neither did, but Joe's reply was to caption the photo, "Partnerships, the old and the new." And with that, I thank you for your attention. I'm happy to raise a glass to both Bob and Toby together with all of you.